This is lab 8 and 9, your muscle structure and then muscles of the human body. First model we have is your muscle anatomy. This is one cross-section of one individual muscle cell. A muscle cell is referred to as a muscle fiber. Okay? This muscle fiber is going to be made up of smaller fibers. These are myofibrils, the smaller fibers that run the length of the muscle fiber. It will have its own plasma membrane called the sarcolemma. It will have a nucleus, okay? And mitochondria are associated here, these small ones, all located in here. Structures found only in the muscle fiber that we've talked about in lecture. We have what's called the sarcoplasmic reticulum. This is found in all the cells, but it's special in the muscle fiber in that it's a calcium reservoir. That is this tan structure. And the tan structure is surrounded by T-tubules. T-tubules come in from the surface. They bring down the electrical impulse surrounding the sarcoplasmic reticulum and will strike the fatter ends of the sarcoplasmic reticulum. We have two of these, one on either side of the T-tubule. These fatter areas are called terminal cisternines, and they are where the calcium is stored. So when the electrical impulse is brought in, it hits these two terminal cisternines, releases the calcium. This group of three, terminal cisternine, the T-tubule, which is the blue, and the terminal cisternine on the other side, is called the triad, because this is where all the activity is. Okay. In your myofibrils, which are these strands that run the full length, they have banding patterns, and it's going to go from a dark A band to a light I band to a dark A band. Okay? And these bands will have different zones within them, not easily seen on this model, but when we look at the the sarcomere model, we will. A sarcomere is a repeating unit from the middle of the I band to the middle of the next I band, little light colored bands. And coming from them, we're going to have two different proteins. We're going to have thin actin going across in the I band, and we're going to have a thicker myosin protein in the middle here, which makes it a darker color because of the myosin. Okay? And these sarcomeres are from the middle of the I band to the other, are attached to what are called the Z disc. Okay? Now, the next model we're going to look at is big sarcomere. Now, your sarcomere is one of those repeating units. This covering here is got the yellow T-tubules. The green is the sarcoplasmic reticulum. This area is the thicker terminal cisternine. Here's another thick terminal cisternine. This group of three between my fingers, terminal cisternine, T-tubule, terminal cisternine, is called the triad. Tri means three. When we peel this off, we can now see the structure of a sarcomere. A sarcomere is a single repeating unit of the muscle. These are both the ends. These are the Z discs or Z lines. They'll be in the middle of what we call the I band, the lighter colored band. Okay. Coming from your Z line are the protein actin. Okay, that's what these fibers are. They go towards the middle, not all the way into the middle. In the very middle, we have the M line, M for middle. And coming off of that is our thick, dark purple protein, the myosin, which has little heads on it that will allow it to reach up and grab the actin and bring it in. Now, with the sarcomere, we have the M line is in the middle of the dark band. The dark band is from one end of the myosin to the other end of the myosin. Okay? That dark band is called the A band. Dark has the letter A in it. Okay? So the dark band is myosin to myosin. It's got the M line in the middle. And then where there is the zone where there is only the myosin, no actin at all, in this portion of the dark band only, this is called the H zone. Where there is both myosin and actin, it's a zone of overlap. So it's going to go from here to here, and we're going to have a second zone of overlap over here. The I band, the light band, is going to go from the end of the myosin, okay, to the other end of the myosin in the next sarcomere, and right in the middle of that is the Z line. Okay. What will happen quickly is that you will have the actin reach up and grab the myosin. The myosin reach up and grab the actin and start pulling the actin inward, pulling the Z line in and causing the muscle fibers, sarcomeres, to contract. When they all do this at the same time, the whole muscle fiber contracts. And when many muscle fibers are doing it at one time, it will cause some degree of muscle contraction. Okay. The other thing you have to make sure you look at is that we have one slide on this model, on this lab, and this slide is the same skeletal muscles picture you saw before, but you have to identify three things: the nucleus, the A band, which is the dark repeating bands, and the I band, which are the light repeating bands. Dark has the letter A, light has the letter I in it to help you remember ways of identifying those bands. Now, also in your handout, it tells you to look at some figures in the book, to look at the different connective tissues associated with the muscle, epimysium, endomysium, and make sure you look at those figures because those are also possible for the lab practical.